In this video, we will look at the use of paleogenetics, which is the extraction and analysis of ancient DNA, to understand the origins of the people of India. The contents of this video are based on a website that I created on this topic. The website is available at https://sites.google.com/view/ourorigins/home. This presentation is provided for educational and informational purposes only. The data is gathered from various external sources. Their inclusion does not constitute an endorsement of the original authors, their viewpoints, or their methodologies. The contents should not be construed as a criticism, negative evaluation, or disparagement of any individual, their opinions, their beliefs, any organization, any government, or its departments. Every effort has been made to ensure the accuracy of the data presented. Any inadvertent errors or omissions are unintentional and will be corrected upon receipt of verified data. The audio has been generated using Google TTS. Some name and scientific terms might be mispronounced. Your understanding is appreciated. In the previous video, we identified the key disciplines used to trace the origins of the people of India. They were archaeology, anthropology, radiometric dating, genetics, and linguistics. In this video, we will focus on paleogenetics, which is the analysis of ancient DNA. We will start with an overview of cells, chromosomes, DNA, and genes. We will define the concept of haplogroups and haplotypes with focus on mtDNA haplogroups and Y chromosome haplogroup. The use of haplogroups to understand the most recent migration of Homo sapiens will be examined in detail. Note: the figures in this video are from Wikimedia Commons or generated using AI tools or have been created by me. The cell is the smallest fully functional unit of a living organism. It has a membrane, cover, that encloses the various parts of a cell, such as the nucleus, cytoplasm, mitochondria, ribosomes, etc. A human body has trillions of cells. The nucleus of a cell has 23 pairs of chromosomes. Chromosomes are structures that contain and organize the DNA of an organism. DNA, deoxyribonucleic acid, is a long molecule that carries genetic information and is passed from parents to children. DNA is the molecule that carries the genetic information used for the growth, development, and functioning of living organisms. This figure shows a human cell, the nucleus, the structure of the DNA, the 4 bases in the DNA, and the sugar-phosphate backbone of the DNA. The 4 bases are called adenine, guanine, thymine, and cytosine. Adenine binds only with thymine, and guanine binds only with cytosine. The 46 chromosomes in the nucleus of a human cell together constitutes most of the individual's genomic DNA. The genome is the complete set of genetic material in an organism. There is a small amount of DNA, about 0.1%, outside the nucleus, in the mitochondria. This is called mTNA and plays an important role in tracing ancestry. Almost every cell in a human carries the entire genome of the individual. Some exceptions are red blood cells and cells involved in reproduction. The DNA, shown on the right, is in the form of a double helix. Different sequences of the 4 bases form genes, which influence the characteristics of an individual. These images are courtesy of Wikimedia Commons. For each pair of chromosomes, one chromosome comes from the mother, the other chromosome comes from the father. 22 numbered pairs of chromosomes are called autosomes. There is one chromosome pair that determines the gender of the individual. An individual who inherits the X chromosome from both the mother and father will be a female. An individual inherits the X chromosome from the mother and the Y chromosome from the father will be a male. This figure shows the role of the X and Y chromosomes in determining the gender of the offspring. This image has been generated using Google's image generation tool. Ancestry can be traced by analyzing two chromosomes, the Y chromosome and the mitochondrial DNA chromosome. The mitochondrial DNA, mtDNA, is in the mitochondria outside the nucleus. The Y chromosome is passed from grandfather to son to grandson to great-grandson, etc. The Y chromosome is used to trace ancestry along the patrilineal line. The mtDNA chromosome is passed from the mother to her children, both sons and daughters, but only the daughter passes the DNA to her children. The mtDNA chromosome can be used to trace ancestry along the matrilineal line. A gene is the basic functional and physical unit of heredity and is made up of a sequence of 4 bases, labeled A, adenine, G, guanine, T, thymine, and C, cytosine. It is estimated that humans have 20,000 to 25,000 genes. Each gene is a specific sequence of DNA which gives a certain characteristic to the individual. For example, the hemoglobin beta gene in chromosome 11 makes a component of hemoglobin. The OCA2 and HERC2 genes in chromosome 15 determines the color of the eyes. Sometimes a base mutates, changes, to one of the other bases. For example, A can mutate to G, T, or C. This change is passed down to the following generations. This table shows a few genes, the chromosome that has the gene, and the function of the gene. In some cases, mutations in the gene will cause health problems. For example, a mutation in the BRCA1 gene on chromosome 17 increases the risk of breast cancer. If a mutation is on a sex chromosome, the resultant health issue will be more likely in one gender. For example, the F8 mutation in the X chromosome causes hemophilia in men. Hemophilia is a disease where the clotting of blood is impaired. This does not impact women because they have 2 X chromosomes, and the F8 gene on the other X chromosome can make the necessary clotting factor. This is also the reason for Duchenne muscular dystrophy being more prevalent in men. When a large population has the same mutation in the Y chromosome or mtDNA chromosome, it means they have a common ancestor who first developed that mutation. If this mutation, called a single nucleotide polymorphism or SNP, is seen in multiple generations and is stable over time, it can be used to define a new haplogroup. A haplogroup is a group of people who have a common ancestor and is defined by a specific genetic mutation passed down through generations. For example, the Ket community in Siberia, Russia, have a Y haplogroup called Q, which is defined by a mutation called M242. In this mutation, C mutated to T. It has been determined that this happened about 20,000 to 25,000 years ago. The indigenous people of the Americas also have this haplogroup Q. It is believed that the migration from Siberia in Russia to the Americas happened over the Bering Strait. Another mutation called M3 happened about 15,000 years ago among the native population of the Americas. Therefore, the indigenous people of the Americas have the M242 mutation and a subsequent M3 mutation, proving that the natives of the Americas are descended from the people who lived in Siberia. The Ket people of Siberia do not have the M3 mutation. Therefore, they are the ancestors of the native people of America. This figure illustrates the use of genetic mutation, specifically SNP, to determine ancestry. By examining the Y chromosome of the Ket people who live in Siberia and the Y chromosome of the native population of the Americas, it has been shown that the migration of the Ket community over the Bering Strait about 20,000 years ago populated the Americas. The image on the left has been generated using Gemini's image generation tool. The image on the right was generated using an image generation tool from OpenAI. The Y DNA haplogroups are labeled from A to T. A is the oldest haplogroup and is present only in Africa. The oldest mtDNA haplogroups is L, consisting of L0, L1, L2, L3, L4, L5, and L6. It is believed that only people with the L3 haplogroup migrated out of Africa. Other mtDNA haplogroups are M, N, R, U, etc. If we can trace ancestry by following mutations in the Y or mtDNA chromosomes, is it possible to determine when the mutation occurred? The answer is yes. If we can determine how often a Y or mtDNA haplogroup is created, it is possible to trace back the haplogroups to determine when the mutation occurred. The Y chromosome is large with about 57 million base pairs. This means there is a higher probability of a mutation that can create a new Y haplogroup when compared to creating an mtDNA haplogroup, because the mtDNA chromosome has only about 16,500 base pairs. Studies have shown that a new Y haplogroup occurs once every 2,000 years, while a new mtDNA haplogroup occurs once every 8,000 years. These are approximate numbers. If we now look at the mtDNA or Y haplogroup in any population, such as South Asian, Central European, Siberian, trace their preceding haplogroups, use a value of 8,000 years for the mitochondrial clock and 2,000 years for the Y haplogroup clock, it will be possible to determine when the migration from Africa to the rest of the world happened. This figure shows the Y haplogroups. Y DNA haplogroups are named from A to T and subdivided using numbers and lowercase letters. The oldest haplogroup A did not migrate out of Africa. Its branch BT mutated, called the M168 mutation, to create the CT haplogroup about 65,000 to 70,000 years ago. The CTY haplogroup is the origin of all non-African lineages. While many other paternal lineages, A and B, remained in Africa, only the descendants of the CT lineage successfully populated the rest of the world. CT has subhaplogroups D, E, and CF, which have subhaplogroups D and E and C and F respectively. Haplogroup F is the most prevalent Y haplogroup in the world today. F constitutes over 90% of all non-African paternal lineages. The predominant Y haplogroups in India are H, found among South Indians and tribal people, and considered to be indigenous to India, R1A1, found among North Indian upper castes and thought to be associated with Indo-European migration, L, prevalent in South Indians, and O, found among Indians in the Northeast. The time of origin and sometimes the place of origin of the haplogroups are not precise. There are conflicting claims. The data shown here has been collected from various sources. These are the mtNA haplogroups. L is the oldest mtNA haplogroup, and all the existing mtNA haplogroups descend from this group. Humans with the L0, L1, L2, L4, L5, and L6 haplogroups did not migrate out of Africa. A group of people, one of them a woman with the L3 haplogroup, migrated out of Africa. L3 has given rise to M and N. M generally followed a southern coastal route through India and down into Australia and up to East Asia. N generally moved into Asia and the Middle East and branched into R. Interestingly, Europe has N and R mtNA haplogroups but does not have M. There is reason to believe that migration from the Middle East and/or South Asia populated Europe and parts of Central Asia about 40,000 to 50,000 years ago. This table shows the predominant Y and mtDNA haplogroups in India. H is the dominant Y haplogroup in India. It is considered the Indian marker. It is about 50,000 years old and is more prevalent in the south of the country among people who speak Dravidian languages, such as Tamil, Kannada, Telugu, and Malayalam, and among tribal people. The R1Z93 has higher prevalence in the north and is also seen in people of the steppe region of Central Europe. This is a crucial piece of evidence, and we will discuss this in detail in the next video. M and its subclades, M2, M3, and M6, is the dominant mtDNA haplogroup in India and is about 50,000 years old. It is found all over India in about 60% to 70% of the population. The R haplogroup, not to be confused with the Y haplogroup R1Z93 mentioned earlier, is more prevalent in the North and Northwest regions. Each individual has a single Y-DNA haplogroup, if male, and a single mtDNA haplogroup. However, populations contain many different haplogroups from migration and interbreeding, and individuals have mixed ancestry in their autosomal DNA. We will examine this data in more detail in the next video when we look at the data from 2 papers published in 2019. By studying the mutations in the mtDNA haplogroup and estimating the frequency at which a new haplogroup occurs in the mitochondrial clock, it is believed that every person living outside Africa is the descendant of a woman with the L3 mtDNA haplogroup who migrated out of Africa about 60,000 years ago. This is the reason that 99.9% of the DNA is the same for all humans. The 0.1% difference in the DNA sequence between humans gives rise to the incredible diversity that we see around the world. Humans have very low genetic diversity because there is only one species, and 84 to 85% of the population is a descendant of one individual. Other species, such as chimpanzees, gorillas, and dogs, have much higher genetic diversity. This figure, courtesy Wikimedia Commons, shows the migration of Homo sapiens, based on a study of mtDNA. Study of Y-DNA haplogroups yields a similar picture. A group of Homo sapiens migrated out of Africa about 60,000 years ago and populated the rest of the world. When the Neanderthals and the Denisovans became extinct, sapiens were the only species left on Earth. However, humans today have a small amount of Neanderthal and Denisovan genes in them due to interbreeding. The route out of Africa. 4 routes are possible for migrating from Africa to the rest of the world. Across the Straits of Gibraltar from present-day Morocco to Spain. Across the Mediterranean Sea from present-day Tunisia to Sicily. North from East and Central Africa through Egypt into Sinai and into the Levant, present-day Jordan, Syria, Lebanon, etc. This is called the Northern Route. Across the Bab el-Mandeb at the southern end of the Red Sea, from present-day Eritrea in East Africa to Yemen in the Arabian Peninsula. This is called the Southern Route. There is no fossil or DNA evidence for the first 2 routes from Morocco to Spain or Tunisia to Sicily. The genetic lineages in Europe are younger than the genetic lineages in South Asia. There is higher genetic diversity in South Asia compared to Europe, and an older age for H. sapiens in South Asia. This rules out the northern route from Egypt through the Sinai. This leaves the southern route as the most plausible route for the migration from Africa, by crossing the Red Sea. This is a summary of the evidence of Homo sapiens, based on a study of the Y chromosome and mtDNA chromosome haplogroups. All these dates are approximate and could vary by hundreds or even thousands of years. The migrants crossed the Red Sea to reach Yemen in Asia and followed the coastal route to reach other continents. India and Southeast Asia were populated about 60,000 to 55,000 years ago. China and the steppes of Central Asia were populated about 50,000 to 40,000 years ago. Australia and New Guinea were populated about 50,000 years ago. Homo sapiens crossed the Wallis Sea, which separated Sunda, Sumatra, Java, and Borneo were linked by land due to low sea levels, from Sahul. Australia, New Guinea, and Tasmania were one landmass due to the same reason, and was about 90 kilometers wide. Europe was populated about 45,000 years ago, where they coexisted with the Neanderthals. The Neanderthals became extinct about 40,000 years ago. The Americas were populated about 20,000 years ago. Chile was populated about 15,000 years ago. In the next video, we will examine the two papers published in Science and Cell in 2019 that analyzed ancient DNA of the people who lived in India, Pakistan, Iran, and the Tehran region thousands of years ago. We will look at the genetic, linguistic, and archaeological evidence to understand the origins of the people of India.